A book lung is a type of respiration organ used for atmospheric gas exchange that is found in many arachnids, such as scorpions and spiders. Each of these organs is found inside an open ventral abdominal, air-filled cavity, atrium, and connects with the surroundings through a small opening for the purpose of respiration. The oldest book lung found to date belongs to a member of the extinct Trigonotarbida, which lived 410 million years ago and had book lungs already fully adapted to a terrestrial existence. Book lungs are not related to the lungs of modern land-dwelling vertebrates. Their name describes their structure: stacks of alternating air pockets and tissue filled with hemolymph, the arthropod equivalent of blood, give them an appearance similar to a folded book. Their number varies from just one pair in most spiders to 4 pairs in scorpions. The unfolded pages, plates, of the book lung are filled with hemolymph. The folds maximize the surface exposed to air, and thereby maximize the amount of gas exchanged with the environment. In most species, no motion of the plates is required to facilitate this kind of respiration. Sometimes, book lungs can be absent, and gas exchange is performed by the thin walls inside the cavity instead, with their surface area increased by branching into the body as thin tubes called trachea. The trachea possibly have evolved directly from the book lungs because, in some spiders, the trachea have a small number of greatly elongated chambers. Many arachnids, such as mites and harvestmen, opiliones, have no traces of book lungs and breathe through trachea or through their body surfaces only. The absence or presence of book lungs divides the arachnida into 2 main groups: the pulmonate arachnids, book lungs present, scorpions, and the tetrapulmonata, whip scorpions, schizomeda, amblypygi, and spiders, and the apulmonate arachnids, book lungs absent, micro-whip scorpions, harvestmen, acarina, pseudoscorpions, Riccinulae, and sun spiders. One of the long-running controversies in arachnid evolution is whether the book lung evolved from book gills just once in a common arachnid ancestor, or whether it evolved in multiple groups of arachnids in parallel as they came onto land. The oldest book lungs have been recovered from extinct Trigonotarbid arachnids preserved in the 410-million-year-old Rhynie Church of Scotland. These Devonian fossil lungs are almost indistinguishable from the lungs of modern arachnids. Book gills. It is believed that book lungs evolved from book gills. Although they have a similar book-like structure, book gills are found externally, while book lungs are found internally. Book gills are still found in horseshoe crabs, which have 5 pairs of them, the flap in front of them being the genital operculum, which lacks gills. Book gills are flap-like appendages that affect gas exchange within water and seem to have their origin as modified legs. On the inside of each appendage, over 100 thin leaf-like membranes, lamellae, appearing as pages in a book, are where gas exchange takes place. These appendages move rhythmically to drive blood in and out of the lamellae and to circulate water over them, respiration being their main purpose. They can also be used for swimming in young individuals. If they're kept moist, the horseshoe crab can live on land for many hours. In the marine arthropod Limulus, the book gills are developed from the base of the abdominal or opisthosomal appendages. These 5 pairs of appendages are flap-like and membranous, with the undersurface of each flap formed into many leaf-like folds called lamellae. Thus each gill bears hundreds of thin membranous lamellae arranged like pages in a book. Each lamella is provided with capillaries separated from the outer sea water by a thin wall that acts as the osmoregulatory membrane. The movement of the organs maintain a constant water flow for gaseous exchange. The oxygen is diffused in from the water and the carbon dioxide is expelled out. The cocci of the last pair of appendages bear short stapulate flagella, which helps to clean the gills and serves to sense the oxygen content of the water current.